Okay, so in our previous lecture, we talked in detail about carbohydrate catabolism. Now we're going to talk about how we can use the energy from other macronutrients. So we're going to start with fats, and specifically we're going to talk about lipolysis. Lipolysis is, we're ending with this word lysis, which we know cues us into the fact that this is a catabolic or breaking down reaction. In the process of lipolysis, we are going to start with our triglyceride. So remember, our triglyceride has this glycerol backbone and 3 fatty acid tails. In the process of lipolysis, we are going to cleave off fatty acids off of this glycerol backbone. And so that is what we see here. We have cleaved off fatty acids from the glycerol backbone. And these fatty acids are then going to travel from the cytosol into the mitochondria in order to go through another pathway of chemical reaction so that we can synthesize some ATP. And then this glycerol backbone that was formerly part of the triglyceride is going to get shuttled towards glycolysis. So we can also break down this glycerol backbone in order to also generate some energy and some reduced compounds. Okay, so here is our fatty acid, and we are now inside the mitochondria, inside our mitochondrial matrix. The process that we use, the series of chemical reactions that we are going to use in order to break down this fatty acid is called beta oxidation. And in the process of beta oxidation, we are going to go through a series of chemical reactions that are essentially going to cleave off 2 carbons at a time from our long fatty acid tail. And so it's a whole series of reactions that I'm just representing with this one arrow, but at the end we end up with a 2-carbon group that was originally from the fatty acid, and it's going to be attached to coenzyme A to make acetyl-CoA. Now, acetyl-CoA may be familiar, it may be triggering your memory because it was the end of glycolysis. Remember, glycolysis ended with pyruvate that then got converted into acetyl-CoA to go into the Krebs cycle. So this should be familiar. Now, in the process of cleaving off these two carbon units off the fatty acid, we're going to reduce some NAD to NADH. We're also going to reduce some FAD to FADH2. So we're going to generate some reduced compounds in the process of beta oxidation. And then additionally, we are going to have this coenzyme A, CoA, that is going to get added to the 2 carbons that got cleaved off of our fatty acid tail to yield acetyl-CoA. Now, something else that I would like you to appreciate is that look at how many 2-carbon units we have in this fatty acid. And fatty acids can come in much longer lengths than this particular one. So we just think about how in fatty acids, we have so many opportunities to cleave off 2-carbon units that we then shuttle into Krebs cycle and can use to synthesize ATP. And then from there, this acetyl-CoA is going to enter the Krebs cycle, just like I did when we were talking about in the last lecture after glycolysis. And so that acetyl-CoA is going to enter the Krebs cycle, go through all of those same reshuffling, rearrangements, and ultimately it's going to help us reduce a whole bunch of NADH. We're going to get a whole bunch of reduced NADH. So 3 NADHs. We're going to reduce some FAD to FADH2. We're also going to generate a little bit of ATP. And all of these reduced compounds, all of the NADHs, all the FADH2s, those are all going to get shuttled towards the electron transport chain, just like we talked before. And between the electron transport chain and ATP synthase, we are going to be able to synthesize ATP. So remember, just like we talked about in the previous lecture, all of those hydrogens from the reduced NADHs and FADH2s, they are going to arrive at the electron transport chain. That electron transport chain is going to rip the hydrogens apart into a proton and an electron. The protons are going to be pumped into this inner membrane space, and the electrons will bounce down the electron transport chain. And then the ATP synthase protein complex can harness that potential energy from the high concentration of protons that are in this inner membrane space in order to synthesize ATP, in order to add a phosphate to ADP and ultimately synthesize ATP. So that part is all exactly the same. So overall here, the process of beta oxidation is going to cleave off 2 carbons at a time from our long fatty acid tail, ultimately to yield some acetyl-CoA. From there, that acetyl-CoA will go through the Krebs cycle and produce a whole bunch of reduced compounds that then go through the electron transport chain and allow us to synthesize ATP.